Hello future doctors, welcome to Dipenism. I am Dr. Dipen Shah and today we are going to discuss about the topic of enzymes. In fact, my first video was on enzymes itself, and from that day till the present day, the journey has been wonderful, and thanks to all of you. And because of the request of all the future doctors, I am again continuing with this topic of enzymes, where we are going to discuss about a specific topic, that is the factors which are controlling the enzyme activity. In my previous video on enzymes, I have discussed about the chemical properties and the mechanism of action, how enzymes function. And in this video, we are specifically focusing on the factors. I would request all the viewers that when you watch a video, go through my playlist so it becomes easier for you to follow the topics. Otherwise, there might be a bit of randomness in the videos. So please follow the playlist. It becomes easier for you to take the reference. So when we are discussing about the factors affecting enzyme activity, मुझे आप लोग को देना है एक particular tip. In fact, मुझे देना है आप लोगों को tips. So there are tips which are the factors controlling enzyme activity. From T, it stands for temperature. I stands for inhibitors. P stands for pH. And S stands for substrate concentration. So these are the four major factors which controls the enzyme activity. Let me first discuss with temperature and pH, because दोनों का the effect on the enzyme activity is somewhat similar. So when we consider the first two factors, that is temperature and pH, and for considering both the factors, when we plot a graph wherein on the x-axis we either take temperature as one of the scale, or we take pH, and on the y-axis we take the enzyme activity or the rate of reaction on the y-axis. And when we plot a graph, then the graph shows a particular characteristic feature, that is, whenever the temperature is low, even the enzyme activity is low. Whenever the temperature is extremely high, still the enzyme activity is low. So at low temperature and extreme high temperatures, the enzyme activity remains low. The enzyme activity increases with increase in temperature, and specifically at moderate temperatures, the enzyme activity is the highest. So the graph appears like this, wherein at low temperature there is enzymes becoming inactive or inhibited, and at extreme high temperature what happens is there is denaturation of the enzyme, because enzymes are specifically proteins in nature. So at high temperature what will happen is there will be denaturation of the proteins. So the amino acid which are forming the bond, that will be destroyed and enzymes will be totally denatured. So only at moderate temperature the enzyme activity is the maximum. So when the temperature increases, the enzyme activity also increases, but after further increase, the enzyme activity again decreases. So that temperature, which is the optimum temperature, that is known as temperature optima, which is usually the body temperature. That is why हम लोग खाने की चीज़ों को रखते हैं रेफ्रिजरेटर में, दैट इज एट लो टेम्परेचर, सो दैट बैक्टीरियल एंजाइम या दूसरे माइक्रो ऑर्गेनिजम्स के एंजाइम्स जो होंगे दैट विल गेट इनहिबिटेड. Even after removal from the refrigerator, the food has to be cooked or heated so that at high temperature वो enzymes totally denature हो जाए and destroy हो जाए. So that is the reason why we refrigerate and heat the food, so that bacterial activity and their enzyme activity gets totally inhibited or destroyed. The similar thing also appears for the pH. At low pH and high pH, the enzyme activity is going to be low. Only at moderate pH, which is known as the optimum pH, the enzyme activity is going to be the maximum. So that pH we usually consider that is around the neutral pH. Yes, there are specific enzymes जो function करते even at low pH, that is at acidic pH. We know, okay, in the stomach the HCl secretion maintains the acidic pH, जो allow करेगा the enzyme pepsinogen to be converted into pepsin. So there are certain enzymes which can function at low pH. There are certain enzymes which work at alkaline pH as well. जैसे कि small intestine में the enzymes of the pancreas, they mainly function at alkaline pH. So there are certain enzymes which can function at alkaline or acidic pH, but usually most of the enzymes function best at the moderate pH, that is known as the pH optima as well. So this we have discussed the two factors, temperature and pH. Let's move to the next factor, that is the substrate concentration. So let's discuss the third factor, that is about the substrate concentration. So suppose I am representing over here particular enzymes. So I am representing total 3 enzymes, and these 3 enzymes have each having single active site. So there are total 3 active sites that are present with these 3 enzymes, and suppose these are the substrates which can bind with the active site of the enzyme, and they will be converted into the products. So the first substrate binds with the enzyme's active site and rate of reaction is going to increase. Substrate concentration further increases, it can bind with the another enzyme's active site and rate of reaction further increases. Substrate concentration और further बढ़ेगा, enzyme activity increases. Substrate concentration और बढ़ रहा है, no effect, और बढ़ रहा है, no effect, और बढ़ रहा है, no effect. So what we understand is, at a particular point, the rate of reaction reaches the maximum point, wherein all the active sites of the enzymes, once they get saturated, then increase in substrate concentration further does not increase the rate of reaction. So when we plot a graph representing the enzyme activity with the substrate concentration, then as the substrate concentration increases, the enzyme activity also increases, but up to a particular point, after which the increase in substrate concentration does not increase the enzyme activity, and enzyme activity reaches the maximum point, which we call it as Vmax, or that is also known as maximum velocity of a reaction. So after this point, even if the substrate goes on increasing, the enzyme activity is not going to increase because the active sites are saturated. Once the active sites again becomes free, that means the substrate product में convert हो जाएगा, फिर active site का availability हुआ, again the rate of reaction may proceed. So it all depends on the availability of active sites. इसीलिए many people when they consume lot of food, still उन पे कोई effect नहीं होता है. There is no weight gain. Now the reason for that is it all depends on your enzymatic activity. Beyond a limit हम लोग substrate ज़्यादा बढ़ाते भी जाएंगे तो भी it is not going to convert into product. It is finally going to remain as undigested food. So you have to understand the metabolic reaction of a person, and based on that the diet plan is suggested. So we have discussed 3 factors, that is temperature, pH and substrate concentration. Moving to the 4th important factor. Now the next factor is the inhibitors. Now inhibitors are certain chemicals or substances which inhibits or shuts off the activity of enzyme. So such chemicals are known as the inhibitors, which are going to stop the activity of enzymes. So these inhibitors, they show 2 different types of inhibition mechanism. Let us understand the first mechanism. Suppose यहाँ पे मैंने represent किया है the enzyme, and this site is the active site. इस active site पे a particular substrate can come and bind and it can be converted into a product. लेकिन suppose इस substrate के जैसा एक similar design वाला एक particular chemical है which can come and bind with the active site, and it will not allow the binding of the substrate to the active site, then this chemical substance is going to be called as the inhibitor. So यहाँ पे what is happening is the inhibitor is competing for the active site with the substrate. Hence, this chemical is going to be called as a competitive inhibitor. And this mechanism is known as competitive inhibition. So in the inhibitor part, we have discussed the first type of inhibitor, that is a competitive inhibitor, जो chemical active site के लिए compete करेगा for the against the substrate. Since ये inhibitor इस active site पे bind हो पा रहा है, the reason for that is इसका design या इसका structure is similar to that of the substrate. Hence, this inhibitor is known as substrate analog. So it is basically similar in structure with the substrate. There are certain specific examples of competitive inhibitors, and one of the example is an inhibitor known as malonate, which is a competitive inhibitor of a substrate succinate. So in this case, malonate is the inhibitor, succinate is the substrate, and ये inhibitor जिस enzyme को stop करता है, that enzyme is succinate dehydrogenase. Now succinate dehydrogenase, this enzyme is the enzyme present in the Krebs cycle, wherein इस enzyme से succinate substrate bind होगा and it is converted into the product fumarate. But in presence of this competitive inhibitor malonate, which is a substrate analog of succinate, malonate can bind with the enzyme and it can inhibit the activity of succinate dehydrogenase. So what will happen is this will stop the event of proceeding of the Krebs cycle. We usually use malonate as a chemical inhibitor to kill the unwanted plants, that is the weeds. So it can be used as a weedicide as well. Another example is, suppose this enzyme that is present, it is a bacterial enzyme, and this bacterial enzyme को for synthesis of a particular product folic acid, that is for folic acid synthesis, it requires a substrate which is known as PABA, that is known as para amino benzoic acid. So understand this mechanism जहाँ पे para amino benzoic acid is the substrate. This binds with the enzyme and it will be converted into the product folic acid. तो ये folic acid bacteria के लिए ज़रूरी है for its survival. Now, in order to kill the bacteria, what we do is we use certain chemicals which is going to be the substrate analog of PABA. And such chemicals which we have used are the sulfa drugs. Now these are the medicines which are acting as inhibitor or substrate analog of PABA. It will inhibit the enzyme activity. So what will happen is para amino benzoic acid bind नहीं कर पाएगा and folic acid का synthesis stop हो जाएगा. And that is how, since folic acid नहीं बनेगा, ultimately the bacteria's life cycle is going to get inhibited and ultimately bacteria का death हो जाएगा. But remember future doctors कि ये competitive inhibition जो होता है, this is a reversible mechanism. That means substrate concentration अगर बढ़ जाए, then in that competition substrate will win and inhibitors का activity will be stopped. So this is a reversible mechanism. Hence it is important कि हम लोग जब भी medicines patient को देते हैं, it is given for particular time duration, जहाँ पे we give the particular antibiotic 3 times in a day, 2 times in a day, so that उसका inhibitor का concentration हमेशा up to the mark रहे and वो substrate वो बाइंड ही ना कर पाए एंड बैक्टीरिया का डेथ हो जाए. एंड दैट इज व्हाई वी स्प्रेड द डोज ओवर अ टाइम ड्यूरेशन ऑफ 3 days or 5 days so that the concentration of that medicinal drug is kept high. So this we have discussed about one type of inhibition, that is competitive inhibition. Let us discuss another type of inhibitors. Now in that another type of inhibitors, the concept revolves around another site as well of the enzyme. This another site of the enzyme is known as the allosteric site, and such enzymes we call it as allosteric enzymes. This was the active site of the enzyme, this is the allosteric site. Now in this type of inhibition, what happens is the inhibitor does not compete for the active site. Instead the inhibitor comes and binds at the allosteric site and it changes the configuration of the enzyme in such a way कि उसका inhibition हो जाता है. So here the binding does not occur at the active site, it occurs at the allosteric site. Hence this type of inhibition we call it as non-competitive inhibition. and the inhibitor is known as the non-competitive inhibitor. A particular example in this case is of certain toxic metals which act as non-competitive inhibitor, or CO, that is carbon monoxide, or CN, that is cyanide. Now these inhibitors, what they do is they come and bind at the allosteric site of the enzyme and active site का structure totally destroy कर देते हैं. What they do is they destroy the sulfhydryl groups which are present at the active site, which are also known as SH groups, and because of the total destruction of the active site, now the substrates cannot bind at the active site. So enzyme activity has been destroyed permanently, hence this inhibition is an irreversible mechanism. This is an irreversible non-competitive inhibition. One of the enzyme that is inhibited by carbon monoxide or cyanide is the enzyme cytochrome C oxidase. Remember future doctors, this is a very important enzyme जिसका रोल होता है in electron transport system which occurs on the oxysomes, and ये enzyme अगर inhibit हो गया तो ETS का process stop हो जाएगा, ATP release बंद हो जाएगा, it can result in death of the patient as well. So carbon monoxide, cyanide, these are non-competitive inhibitors which irreversibly destroys the enzyme activity. The third type of mechanism which is non-competitive, but in that case that is reversible, that has to be discussed. So far we have discussed about the competitive inhibition that was reversible. We have discussed about non-competitive inhibition which was irreversible. Now coming to the last type of inhibition, which is again non-competitive, but that is of reversible type. Now in this mechanism we have to understand the activities of two enzymes. Now both the enzymes are related with the process of glycolysis. I already made one video on glycolysis, you can go through that video जिनको glycolysis का process नहीं पता, wherein glucose gets converted to glucose 6-phosphate, glucose 6-phosphate is converted to fructose 6-phosphate, and fructose 6 is converted to fructose 1,6-diphosphate. Now in the first step the enzyme involved is the enzyme hexokinase, and in the third step the enzyme involved is phosphofructokinase. So this mechanism that we are discussing right now, it is mainly based on these two enzymes. So let us begin with the concept wherein suppose this enzyme is the enzyme that is hexokinase, and on this enzyme hexokinase, which is the active site, what will come and attach is the substrate. That substrate is the glucose. Once glucose binds with the active site, वो active site पे आके convert हो जाएगा into the product, and the product in this case is glucose 6-phosphate. Now understand future doctors कि ये जो product form हुआ है from this substrate, the product that has been formed, ये substrate goes and binds with the allosteric site, and allosteric site पे जाके वो कॉन्फ़िगरेशन इस हिसाब से चेंज कर रहा है कि एक्सोकाइनेज को इनहिबिट करता है फॉर फर्दर कन्वर्जन ऑफ ग्लूकोज इनटू ग्लूकोज 6-phosphate. सो यहाँ पे द ग्लूकोज 6-phosphate प्रोडक्ट इज एक्टिंग एज अ इनहिबिटर. सो हियर व्हाट हैज हैपेंड इज प्रोडक्ट ने ही एंजाइम एक्टिविटी को रोक दिया है, हेंस दिस इज नोन एज प्रोडक्ट इनहिबिशन. ऑल्सो this product inhibition is basically conversion of the substrate into the product. So basically एक response दिया एंजाइम ने substrate को product में convert करने के, and ये response जो है which is also known as a feedback. So यहाँ पे जो feedback मिला है product का, that feedback is itself acting as a inhibitor, hence it is also known as feedback inhibition. या फिर product ही खुद जाके उल्टा जाके एंजाइम को रोक रहा है, तो ये पीछे जाके एक एक्टिविटी कर रहा है, प्रोसीड नहीं कर रहा है आगे, हेंस इट इज ऑल्सो नोन एज रेट्रो इनहिबिशन. सो दिस इज वन टाइप ऑफ non-competitive inhibition wherein the activity is reversible. Because जैसे ही glucose 6 phosphate convert हो जाएगा into fructose 6 phosphate by the activity of another enzyme phosphoglucoisomerase. So once this product which is acting as inhibitor in this case, जैसे ये convert हो जाएगा into the next chemical, ये activity अपना stop कर देगा. Hexokinase will again become ready and glucose वापस से bind हो सकता है and glucose 6 phosphate में conversion होगा. Again the cycle will continue. Hexokinase वापस inhibit हो जाएगा, but that is again reversible. Now let us understand कि enzyme phosphofructokinase कैसे अपना activity दिखाता है. So for the enzyme phosphofructokinase, what happens is reactant fructose 6 phosphate comes and the substrate gets converted into fructose 1,6 diphosphate. But here the product are not playing any role in the inhibition of the enzyme phosphofructokinase. Instead, it is the ATP molecule which inhibits the enzyme. Now what happens is ये glycolysis, Krebs cycle ये सब event होता क्यों है? So that ATP का generation हो सके. But in case ATP का availability already है, then why should an enzyme keep on metabolizing all the substrates? So what happens is when ATP is present, it does not allow the phosphofructokinase enzyme to function. एंड वो इस हिसाब से इस एक्टिविटी को मॉड्यूलेट करता है कि इट गिव्स अ नेगेटिव इफेक्ट ऑन द एंजाइम फॉस्फोफ्रक्टोकाइनेज एंड इट स्टॉप्स इट्स एक्टिविटी. सो हियर एटीपी इज एक्टिंग एज अ नेगेटिव मॉड्यूलेटर, बट दिस इज अ रिवर्सिबल प्रोसेस. वंस अगर एटीपी का यूटिलाइजेशन हो गया एंड एटीपी इज ब्रोकन डाउन इनटू एडीपी और इनटू एएमपी because of the hydrolysis and energy का release होगा. So now अभी ATP generate करना पड़ेगा वापस. So what will happen is this ADP and AMP they go and bind with the allosteric site. So this can be ADP or AMP and ये जो chemicals है ये phosphofructokinase को positively activate करते हैं. They positively modulate. So they are known as positive modulators जो phosphofructokinase को अलाउ करेगा कि इस सबस्ट्रेट का कन्वर्जन हो सके इनटू द प्रोडक्ट. सो हियर दिस इज अगेन अ टाइप ऑफ नॉन कॉम्पिटिटिव इनहिबिशन जहां पे एलोस्टेरिक साइट पे इनहिबिशन हो रहा है. बट हियर द रेगुलेशन कैन बी इधर पॉजिटिव मॉड्यूलेशन और नेगेटिव मॉड्यूलेशन विच इज बाय टू डिफरेंट केमिकल्स. तो यहाँ पे सिर्फ इनहिबिशन ही नहीं हो रहा है, हियर देयर कैन बी एक्टिवेशन ऑफ द एंजाइम एज वेल, बट दैट इज अगेन एट द allosteric site, so it is again non-competitive. So this overall we have discussed the 4 factors which control the enzyme activity, wherein the major focus for your NEET and AIIMS exam is on the concept of inhibitors. Stay tuned for my further videos, and that's all from Dipenism.